Good morning, everyone.
Good morning.
Thank you. So why don't we start with a couple of comments. Some of the math I did at the end with diffusion was a little confusing, and I appreciate that because I had to go through it very quickly. The good thing is there's some homework on it. So this person was confused about many things. I'm glad there's homework on this. You will have another homework assigned on Thursday, and it will be due probably the next Thursday or maybe Tuesday after that. And I'm sorry to say that will be the last homework. So, I know. Yes, it's not due on Thanksgiving. Although if anyone wants to send it as a Thanksgiving present to me, I will not accept it. Okay. Although this is sort of irrelevant, given this person had a fair amount to complain about in terms of the homework. He or she says greater than 10 homeworks is not fair. Okay? Next time I'll say that there's 11 or 12, or maybe I'll say there are 13. Then they'll say, oh, how nice. There's only 12. Can you give more time for homeworks, as the last one was quite intense? It's true, it was intense. Can I give more time? Well, and this, I want you to think about this, because I want you to come up with an answer. In very general terms, what makes swimming on a microscopic scale different than on the macroscopic scale? That is an excellent question, because what you have to do is have to think about everything and then summarize it such that, you know, a second grader could understand it. Because it clearly is very different. How is it very different? Yes?
Instead of just kind of moving through like atoms and molecules and like pushing against them, you're kind of like pushing off stuff and going through a little vacuum until you hit the next molecule or atom.
Yeah.
I mean, since we were talking about when you bump into the other molecules, so moving into the vacuum, so that'd be weird.
Okay, that's very close. Tell me about I want you to think about it. I want you to do this in a hurry because I want you to think about it now. And in 2 minutes, you're going to give me a response. Explain how it's very different in a macroscopic sense. A bacteria is swimming along and then, for example, shuts off its motor, versus you being macroscopic are swimming along and then you shut off your motor. How is it very, very different? So what's it like for a bacteria swimming and what's it like for you swimming? And I don't want any math involved, but just the conclusion. Although it's similar to what she said, but I think if you're in second grade, you still might have a little bit of difficulty with it. So go ahead, 2 minutes, think about it.
And then you stop swimming all the time.
Yeah. Isn't that a good question?
It's one that I'm like, there's some similarities.
No, I agree.
Usually we have to,
I'm just listening.
yeah, like 10,000 molecules hitting one bacteria, 10,000 molecules.
Microscopic is difficult, right?
What was that?
Microscopic swimming is difficult.
I don't think this second grader is going to understand what half a KT is.
Yeah, but it's not so much. No.
So what does it mean in plain English? Are you two talking?
Yeah, we talked about.
You did?
We think we. Yeah.
Oh, yeah.
No.
Okay. So, wait, let's see. A second or third grader, I guess he or she is too young to cry. I want to see, what does the kid like to see? Star Wars?
Yeah.
Yeah, okay.
Mickey Mouse.
Oh, good, Mickey Mouse. I want to see Mickey Mouse. So for some reason, I'm supposed to listen to you explain about why, how a bacteria swims versus you swim. So tell me a story. It shouldn't involve things like KT and 1/2 mv squared. Go ahead. Tell me a story. You've tried already, or more than tried. Yes.
So once there was a bacteria.
Once there was a bacteria. Okay, good.
So he was in a large pool of water, but that water was very big. It was size of his body, and it was bouncing on his body from here. He didn't know where to go. So he just went to sleep and then let them drive his motion.
Oh.
And he did just fine.
Hey,
That was like a good thing.
give him a break. Go ahead.
Yeah. So once there was a man, he wanted to swim, but he couldn't go to sleep.
Come on up here. This is a better story than I thought. Come on.
So once there was a man, he couldn't go to sleep because nobody will bounce him and drive him along. So he had to do some stuff to swim. So sense of the story is bacteria could go to sleep while swimming. We can't.
Bacteria can go to sleep.
Why could bacteria go to sleep but not people?
Because we won't swim, we will drown. No?
Oh, okay.
That's
Okay. Okay, that's good, sort of. Okay, somebody give an alternative or whatever story. Come on. Yeah, come on.
Well, I said like, wait, I don't even know if I don't know.
Okay, fair enough. Yeah.
And then like,
Wait, excuse me, Josh, you don't get to play in this game. You don't get to play. Go ahead.
bacteria don't have arms and legs like humans do. So that creates a really huge challenge for just swimming around. Like thinking of like not even looking at the fact that they're smaller and they're a little closer to water molecules. And maybe some like viscosity differences might be there, but just the mechanism of trying to swim with no arms or legs is different. So with bacteria, you would have like a flagellum that works a little different than an arm and a leg.
Yeah, it's true. It does work a little bit differently, but we could have a rotor like a submarine or something like that, and that pushes us, and then we stop and we go. Go ahead.
Would you rather have a big car crashing into you or 10 mosquitoes crashing into you?
I don't know about that, because in some senses that's wrong, in the sense of you're relying on the difference in mass between a big car and a bunch of little mosquitoes. And the thing about mass, the MA term, the inertia term, how fast you're going, is not relevant. The thing that you learn is, obviously I wouldn't say this to a second or third grader, but inertia is irrelevant. So that's one of the messages that you guys should get out. Go ahead. Who else? Okay, I'm a little bacteria, and I'm going along. And I'm being pummeled. My big brother or big sister is beating the crap out of me. Oh, you don't see that, but okay. Right? And so I'm trying to run away, but no matter what I do, he keeps pummeling on me. So that's the situation with a bacteria moving, because it has these water molecules which are significant compared to my size. So that causes a diffusion. It also causes, I'm starting to run away, and I may be doing it reasonably well, but if I shut off my motor, these guys are pummeling me really hard. So essentially, immediately, it doesn't matter how fast I was running away. Once I shut off the motor, I'm completely dominated by the pummeling, right? And so it doesn't matter if I times mass times my initial velocity. It essentially immediately I've forgotten about how fast I was going. That says history doesn't matter, or inertia doesn't matter. And it also means that I don't go anywhere as soon as I turn off my motor. That's what's like when you're microscopic. When you're macroscopic, I'm doing my swimming here. Hey, somebody's laughing at my swimming. I can swim not too badly. So-called. You have a comment about my swimming? Do you,
It's very good. I have a question.
my swimming is very good? Okay, so then you can ask your question.
So when we are discussing that bacteria are continuously bombarded by other molecules, that's why he has no control and he is like going wherever they drag him to whatever.
Right.
Like randomly. So it is because these other molecules are comparable mass to the bacteria.
Well, any one molecule is still tiny compared to, huge bacteria. But in fact, there's 10 million water molecules bombarding it. So they are comparable to the or they're greater than the initial inertia that the bacteria had. Okay, so again, when I'm macroscopic and then I'm moving, it's much greater amount of energy than the individual molecules hitting you, and so then you actually go somewhere. Okay? Yes.
So when you mean that inertia doesn't matter, does that
For microscopic.
for microscopic? Okay. So
Right, because for macroscopic, for you and I, it does matter.
right. Okay.
Because you'll turn off your engines, and you remember how fast you were going a second ago. That's what it means for inertia not to matter. Okay? Okay. So, another couple of questions. Will final be during the assigned final exam slot or earlier in the semester? This person apparently missed out. It was like a week ago. It'll be during the final exam spot, which is, I don't know, December 14th or something like that. Okay. This was very nice. Person says, just keep swimming. Yeah, we did calculations relatively quick, yes. You'll have more time during your homework. Okay, so today we're going to switch topics yet again, and we're going to be talking about ion channels. Ion channels are proteins that are in our membranes, and they allow ions, sodium is +1, or calcium, which is a +2, or chlorine, which is a -1. They allow the ions to go through. And they're really important, and I'll get to why they're important. Okay, it turns out, so this is a summary. There are 3 types. One depends on the voltage across the membrane. Another depends on the presence of a ligand, which is basically a small molecule. Whether there's a small molecule there, it causes the channel to open or close. And also there's what's called mechanically sensitive channels. That is, some mechanical thing like pressure forces the channel to be open. And in particular in nerves, which is actually mostly it's a voltage gated, though nerves can also be ligand gated, is they rely on batteries. That is, there's a constant voltage across the membrane, and it's up to nerves to be on or off, whether to make the voltage different from that. The voltage, which we'll get to probably next time, is generated by sodium and potassium exchange. Probably the next time, we'll talk about whether the channel is open or closed, like a transistor, or it's open and then maybe open a little bit more. That is, it's analog. It can be open a lot or little. It turns out that the channel is either open or closed. It's like a transistor. And we'll talk about a little how they figured this out. And lastly, there's analogs in your body which have direct, analog to fruit flies. And it turns out that that's really important, because if you want to do experiments on them, you generally don't want to do them on humans, but you want some sort of model systems. And it turns out there's, for many of these, there's direct examples, and I'll tell you one. So, if I haven't convinced you that these are tremendously important, biologically, I'm going to convince you of that in a second. And most importantly, they will be on the final exam. And so today and next time will be on ion channels, and the last homework will be on ion channels, and this will be important for your grade. Okay, so the reason ion channels are major targets, in this case of psychoactive drugs, is because they are a major target of drugs. We take a drug, they bind to these ion channels, and they turn them on or turn them off, and that makes you feel better. So for example, Valium, which is my favorite drug, nobody finds that funny, okay? It binds to a protein which is which binds ordinarily to a ligand called serotonin. That protein is called the Gaba receptor, and basically it makes the nerve more relaxed, and you feel like, woo, everything's going great. Okay. Oh, and by the way, these are a major source of targets for the various drug industries. Okay, and I talked about there are genes for various ion channels, and one of them controls the amount of potassium. And there are direct analogies, direct mutations that occur in fruit flies that have direct relevance. So this is a woman, and she has difficulty, obviously, controlling her motion. And there's an analogy that if you're interested in studying them, you want to study in fruit flies. Okay, I mentioned that there are 3 types. Voltage gated, which basically means at a certain voltage across the membrane. What is the voltage across the membrane? You've actually gone over this somewhat. So you have a typical cell, and let's say we just define outside being 0 volts, and what is the voltage inside of a cell? Minus 0.1 what? Volts. And so what does this actually, what is it, how do you generate this? Why is this minus 0.1 volts with respect to 0? Come on. Difference in ion concentration. What does it mean? That this is minus and this is, we're saying 0. Is there more plus charges in here or less? Less. Let's say you have some concentration of chloride ion, and then here your chlorine ion is greater. Or you can sodium and is less than outside. Okay? And in the case of nerves, you know, what we're talking about here is like minus 0.1 volts. So in general, there's K plus and some amount of K plus here, and in general, the inside will generally have less positive ions. Okay, so that's voltage gated. Then ligand gated, it turns out, could I have the lights? It turns out that there are some nerves which are ligand gated. There's also your nose, odorants, which are ligand gated, and actually photons. You can think of a photon as a kind of ligand. If there's photons hitting your eye, some ion channel opens up, and you perceive this as seeing. Okay, and finally there's mechanically gated. It turns out in your ear. How do you hear? What happens is there's pressure waves that comes and hits your eardrum, and physically it moves. What it happens, you can imagine like a lawn that, you know, all of a sudden there's a big wind and the lawn, right, will bend over. And in fact, that's exactly what happens, is that you have two like follicles with, it turns out in actin or whatever, some sort of tether, and then this distance actually grows somewhat, and that activates ion channels. The amount of ions ultimately sort of in your brain changes, and that's what you perceive as hearing. Okay? Or another example is if you're a, what is this? What is this called? Venus flytrap. If you have a fly that goes on and with its feet, it touches one of these guys and tends to move it, it sets off some ion channels, which goes, and the fruit fly tends to eat the fly, or the Venus flytrap tends to eat the fly. Okay, and what you find is in E. coli, about 20% of the genes are for ion channels. So if humans is an analogy, right? In humans, there are 20 to 25,000 genes. So in fact, that would mean 5,000 genes, if it's still 20%, deal just with ion channels. So you can imagine it'll keep you busy finding drugs for them. Now in ion channels, there's two types of ion channels. One is called chemical synapses, which in fact is what the one we're primarily going to study. And then there is another called electrical synapse, which we won't study. But all nerves are pretty much chemical synapses that ultimately it changes the chemical. Like before we talked about, it's glutamate, ion channels open, glutamate diffuses across, and that sets up a whole bunch of things. So again, over here, this is the ion channel. Here's the outside, the inside. What you have with ion channels is you have these membrane receptors. This is the case of a ligand-gated. The membrane receptors are normally, let's say, closed, so the ligand can't get through. If you have enough ligand, it manages to open up the pore. The pore being open allows sodium or potassium or chloride and so forth. Okay. So now we're going to study about how nerves work. The thing about nerves is, in general, you have, let's say you have a tickle. And what happens is, at the cell body, you feel something. It isn't particularly a tickle. What happens is commonly like a calcium ion, some plus charge goes in there. What happens is the signal is propagated down an axon, and ultimately it goes into the dendrites, and that ultimately causes you to laugh. So what you have is you have minus about 0.1 volts over here during the normal time. You have your tickle. In fact, what the tickle is again, like a little bit of calcium ions, which cause this excess of plus charges to flow inside. Yes? So what if you're right over here? You're measuring the voltage. The voltage starts to rise. Good. Inside. Then what happens is, let me show. Let me just, we'll get to in a minute how you actually determine what they are, what the voltages are. So you have a plus charge in here. It turns out initially, these ion channels are closed. When the voltage starts going more positive, it tends to make the channels open. So you have more plus charges. It's like you have an avalanche, in that it sees the plus charge, it tends to open even more. So more sodium tends to flow. Yes?
So does the tube in the picture represent like the giant long part of the axon?
Yeah.
Or is this membrane into the cell?
No, I'm sorry. It represents the whole axon. So this is the inside and this is the outside.
And so the red, blue, and yellow, are those ion channels or are those just like biological voltmeters?
There are voltmeters, not necessarily biological. You could go in and stab it right there, and you're measuring the voltage. Initially, the voltage is minus 0.1 volts. You manage to flow some positive ions in. It makes the voltage come up, this would be to 0. As you get the voltage closer to 0, the ion channels tend to open up even more, which allows even more to flow. Now, so what happens? Well, you have some positive charge over here, and then it spreads out. How does it spread out? Well, simply by diffusion, or if you have a big bunch of plus charges, they repel. They spread out. So next to it, it feels a voltage that's more positive than it was just a second ago. But what happens then to the ion channels that are next to it? It tends to open because it's more positive. In that sense, it tends to open, so more positive ions tend to flow. More positive ions tend to flow. It comes in, it diffuses. Next to it, it becomes more positive. And so basically what you have is a little avalanche over here, and then it ends up flowing down. You have this is more positive, and this becomes more positive, and then this becomes more positive. Okay? Yeah.
So, what's the preference of the cell to keep its voltage constant?
Initially, right. Everything's shut off, right? So these ion channels, they're closed, but there's a little bit more sodium on the outside than on the inside. So there's a net minus 100 millivolts.
So, like, let's assume it's minus 100 millivolts.
Uh-huh.
And somehow, because of that tickle, it becomes minus 50 millivolts.
That's right.
And somehow the channel's open.
Yes.
So, if the cell prefers to keep it as minus 100 millivolts, it should like shoot some ions out instead of letting them in. So why it lets them in instead of just like not shooting them out?
These ion channels are here, and for example, in this case, they're sensitive just to sodium. And initially, so they allow sodium in. it turns out it does recover in that eventually, for example, it will allow some potassium to open, that potassium ion channels to open, and then the potassium ion channels allow the potassium to flow in the other direction.
So it just like gets sodium and kicks out potassium because they have like twice more charge and stuff?
No, it doesn't have anything to, it doesn't do anything with potassium initially, because these are just sodium channels. And so it doesn't do anything. It turns out, as we'll see, the fact that ultimately the voltage here becomes more positive does have an effect on potassium channels. And in fact, it opens up the potassium channels and allows the potassium to go outside. Okay, so what we have you have minus, and that this is say, minus 60 millivolts. It could have been minus 100 millivolts. So that's the story. We have a little calcium or a little tickle somehow. The calcium flows in. It makes, because it's a positive ion, it makes this closer to zero. As it gets closer to zero, this is more positive, which tends to make the channels open even more. And eventually, it reaches like a maximum. Then it turns out that the sodium channels decide to close. That is, the thing is open for a certain period of time, but it says, it's enough, I'm going to start to close. And in fact, it does close with a certain time constant. So this starts to fall. It turns out that when this is more positive, all of a sudden there are potassium channels, which sort of look like this but are a little different, which tend to open. So these tend to open. And potassium is very different in that potassium tends to be large concentration here and small concentration over there. So the fact that the channels are open means, just by the gradient, the potassium will flow and will flow this way. Positive ions flowing out, that tends to make the inside more negative, and so it's bringing back the voltage to the initial negative potential. And the timing of this is such that the sodium looks like this, and the potassium looks like that, and the sum of the two will basically look like that. Okay, now Oh. Oh, sorry. Okay, and so it's going to be your job to understand this and to generate it. And basically, it's just what I've said. Over here, the sodium channels, they start to open. More channels, sodium channels start to open. At some point, the sodium channels start to close. And in fact, at that point, also the potassium channel starts to open, and which brings the voltage down. And this can be the sum of these. And in a little bit, you're going to have to do this. Okay, so now what we're going to do is figure out why it's like minus 70 millivolts and then plus 30 millivolts and so forth. Okay, and this is basically a summary of just what I've said. Some positive ions, namely calcium, injected at one side of the nerve causes a local depolarization. It causes sodium channels to open. Sodium flows down its electrochemical gradient. What do I mean by that? Sodium is flowing down its electrochemical gradient. Yes?
There's a high concentration of sodium
Good.
on the exterior of the cell, and
Good.
a lower concentration of sodium within the cell.
Good.
So when the channel is open, it flows down its own concentration gradient into the cell.
Okay, good. That's its chemical gradient. What about its electrical gradient? Why should what about that?
There's also an electric field because of the voltage. It will flow. It'll follow the freedom lines.
Good, good. So both electrical and chemical is going to want to make the sodium flow. Good. When the sodium flows, it causes further depolarization, even a positive polarization. Then the sodium is diffusing inside the membrane. It opens up neighboring sodium, which allows more sodium to enter, causes yet more depolarization, and then you have a net wave of depolarization. Then to close the ion channels, the sodium channels, they spontaneously close. At the same time, the potassium tends to open, causes potassium to flow, and it brings the potential back down negative. Okay. There's a problem with this. Anybody know what it is? There's a problem in terms of, is it going to run out of sodium and potassium? That's what you're saying?
Well, it's gonna run out of potassium.
Why potassium?
Is that what's being released by the...
Okay, yeah. It turns out that in order to generate this kind of voltage, it turns out it requires very few sodium and very few potassium ions. So actually, that's not a big problem. Although your body has a way of regenerating this, in that there is a sodium and potassium pump to bring back the sodium and potassium to what the native levels. So, okay. But there's a problem with this. In terms of, yes,
It has to go in a certain direction.
good. It needs, say it again.
It has to go in a certain direction. So it has to go down the axon towards the synapse.
Towards the dendrite, the other end. That's right. And why is that a problem? Why doesn't?
Because once they go in, initially there's a diffusion to both sides.
Exactly. The diffusion really doesn't care. So what it's saying is you have this initial diffusion, and you should get sodium channels opening up when you're going this way, and you should get sodium channels when you're going this way. How does the cell deal with this? Okay, well, we'll think about it. Okay, yes?
What's the point of the potassium? Why not just pump the sodium back out?
You can do that. You tell me. My father used to do this all the time. It was really annoying. You know, you'd ask questions and like, good point. What do you think? What do you think? Yes? Wait. To pump out potassium. Is it having to pump it out? The sodium is just flowing down its electrochemical gradient, right? Both the electrical and a chemical gradient. What about the potassium? What about its electrochemical potential? That is, if you can rely on that, then let's say this is a potassium. All you have to do is open up the channel and it will flow, just like the sodium was flowing. And if you can do it, that, it turns out, is actually really fast. Why is it fast? It's fast because it's relying on diffusion. And this distance, tell me, what distance is this? How thick is the membrane? How thick is, wait, Josh, you have to give the answer?
I mean, if no one else will, it's about 4 nanometers.
Yeah, it's about 4 or 5 nanometers. I mean, it's really small. So diffusion will do this incredibly fast. So rather than relying on a pump, which means it relies on ATP, it can rely just on diffusion. So interestingly enough, well, it turns out that it has a chemical gradient, because there's a lot more potassium here than over here. So in fact, that is what really drives it. In terms of the electrical, it generally either is neutral or it actually has to fight it somewhat. Okay?
Maybe semi-related question, but after the sodium comes in, is it still more concentrated on the outside? Or does it like equalize?
this is 0. So it actually becomes somewhat more positive on the inside. So there's enough sodium that it actually becomes, instead of minus 60 millivolts, it actually becomes plus. So it actually, okay? Okay. And then here, so you have the wave of sodium. To close the ion, to stop the wave, the sodium spontaneously closes, the potassium ion opens, and it brings the membrane potential back down negative. Okay.
Sorry.
Yeah.
So, like the main question was why is it propagating in one direction, not the other?
That's right.
Are you almost like?
It's a question. That's right. What?
There was no answer to it.
Somehow nature has come up with an answer. So far we don't know it. Okay, let's take a simple system where this is a high potassium and a low sodium. This is in the cytoplasm that is inside the cell, and outside the cell there's a high sodium and low potassium. This is actually what the concentrations are. What happens? Okay, that is here. Oh, this is supposed to be whited out. Okay, so let's say all of a sudden we make this membrane permeant only to sodium. Somehow we're doing that. What's going to happen? So what's going to happen? Sodium's going to flow this way until what point? Okay, to equal concentrations. What is going to happen with the potassium? Huh? Yeah, nothing will happen to the potassium because the membrane isn't permeant. Okay, so what's going to happen to that voltage reading? Is it going to go towards minus 60 or plus 60? Voltage be positive or negative? Which side is 0, actually? You have to decide that. I don't know.
Can we assume now it's 0 because it will show like...
Initially it's 0. And then I guess you'll have more positive over here. So this will read what? Positive. So this is supposed to read plus. Now let's take the situation where the membrane is only sensitive to potassium. Then what happens? Oh, yeah, there it is. See, it was just a little bit delayed. Okay, therefore it'll go to minus. Now the question is, what about, I have plus 59 and minus 59 millivolts based on this various concentration. Why do I say that? why doesn't it go to plus 100 millivolts or plus 500 millivolts or plus 10 volts or something like that? How can we do this? What we know, we already have the information to calculate what this voltage is, because you guys have been sitting in this class. Woo, woo. Okay, I want you to one thing, I apologize, we're not going to have time for a break. Sorry. I know. Yeah, I know. I'm bad. Too bad. I want you to spend the next 3 minutes talking about yourself, and how would you calculate what this is in absolute numbers. You know how to do it. Who can give me an idea, not me, give yourself an idea to figure it out? What do you know? The equilibrium, okay? Go ahead. Come on. Come on. Come on. It's like, what? Yeah, it's a free energy. Right, that's what I want you to get to.
Chemical potential. The concentration of A becomes concentration of B over concentration of A.
Yeah, that's close, but where did that come from? Entropy? Yeah, let's not worry about entropy. What? Who said that? Hey, all right, partition function. What's more simple than that? Partition function is what? What is a partition function? The Boltzmann constant. So you have two states, and you're going to have some Boltzmann constant, and there's some difference in energy between the two states, a plus charge on the outside and a minus, and zero. And so, anyways, go ahead. Monica is here, has given us a start. Go think about it for 2 minutes. How are you going to calculate this? Go ahead, turn it around. Sure, I don't have to answer it.
Yeah, sure. Can we say, okay, like, we know how many protons we have here, how many protons we have here, based on the concentration, and at the equilibrium, we will know like how many protons we'll have, and like the difference between like how many protons will just like move from one side to the other side. We can simply calculate voltage. Now we know it's like 0. If you assume like 0 volt, like delta V would be like trivial. Because you know, like you have this much like protons here, now you have this much proton, like what's the difference with the delta V?
Because I don't know what the frequency is. It's ln
How are you going to get delta V?
in ions. ln.
That's a good question.
I don't know how that relates.
What's the definition of
And then it's
You're talking about.
So, and then, well, one thing about
We use focusing on is that so it's the final concentration, sodium, final concentration.
So, some people are yawning, I see. That's bad. Okay. Does anybody know? Does anybody have an idea? Does anybody give a damn? Okay, what's the Boltzmann factor? Man, you guys are seriously asleep. 1 over the partition function times e to the minus e divided by kt. Probability of being inside and outside. So an ion has some energy outside and some energy inside. So it goes like e to the minus e out minus e in over kt, right? That's the Boltzmann. Okay, so the sodium concentration outside divided by the sodium concentration inside is equal to this. Yeah? That's where you're supposed to all like say yes. Okay, that's good. Okay. So let's just define phi is equal to the voltage. So we have to relate the voltage to the energy. What is the relationship between them? EV, charge times the voltage. On the outside, we say the voltage is 0, just because we have to define it to be 0. So the voltage on, so the E, here, E out is 0, and E in is equal to charge times the voltage inside. So the concentration, this is a plus of in versus out, is equal to minus e to the minus q times phi in over kt. We can take the ln of both sides. The ln of this is equal to minus q times phi in over kt. This, I'm just writing it again. So phi in is equal, So phi in is equal to kt over minus q times the ln. Right there. Q for sodium, what is the sodium? With a plus or minus? Plus, good. Q is equal to plus E for sodium. So KT over, E is just a number. It happens to be 25.7 millivolts. So phi is equal to minus 25.7 times ln of 15 over 150. And that is minus 25.7 times minus 2.3. You know, plug into your little calculator. And so that's equal to plus 59 millivolts if permeable only to sodium. That's good. So if it's permeable only for potassium, the resting potential is what? Remember, is minus 59 millivolts. Okay? So that's good. So, in fact, the resting potential is typically this up to minus 100 millivolts, depending on what exactly the sodium potassium concentrations are. Wait. So this is saying, what is this saying in terms of the cell's membrane? Is it resting? Its resting potential is like minus 60 millivolts. So what is it saying in terms of, is the cell membrane normally permeable to just potassium or sodium or neither? Potassium. Turns out that's true. And is it permeable to sodium? Not at all, except when an action potential is there. Okay, and this is basically the Nernst potential. It's just an example of the Boltzmann constant, that at some point the probability of is 0.5, and it's way down and way up, and exactly where this is depends on the native potassium and sodium. In fact, I'll have you do this. And it actually works amazingly well. Amazingly well. Okay. So I think that will be it. Except the question is, why, when the sodium comes in, why doesn't it flow both ways? The truth is, it does? Does it? No. Sometimes you gotta add a little suspense. So does the sodium, does it diffuse both ways? Yeah, it does.
The overall message could be like to the right.
Yeah. So what's happening? So somebody that I know who's really brilliant said something which actually was totally right. Who could that be? Dr. Josh. Man, I should give him a PhD right now. Josh is saying, yeah, I can believe that. He says that it has to do with inactivation of the sodium channels. Is that right? He nods. How is that working?
That's the part people don't know.
How is that working?
Which sodium channel?
Whatever the sodium channels that were opening. What's that? They're all the same sodium channels. Sodium is sodium. Okay, well,